All right, welcome back to part 7 of DNA replication. Today we are going to explain how DNA replication occurs at the end of eukaryotic chromosomes. Um, Remember that bacterial chromosomes don't have ends, so we didn't have to deal with this problem. We do. Now the ends of our chromosomes are referred to as telomeres. The term telomere refers to a complex of telomeric DNA sequences and proteins bound to it, and together they form a cap at the end of our chromosomes. Now, the telomeric sequences consist of some repetitive um, DNA sequences. So here we see a TTA GGG, TTA GGG, TTA GGG, and you see this happening multiple times. But you'll also find that you'll have a 3' overhang extending about 12 to 16 nucleotides long. These sequences are always rich in guanine and also tend to be fairly thymine rich as well. The actual sequence itself can vary among organisms. So here we have humans, slime molds, various plants, and you can see that the sequence is largely the same, um, with only minor variations. But there are some more diverse things out there. Now, the reason why we need these telomeres is because DNA polymerase can only synthesize in a 5' to 3' direction, and it cannot initiate the DNA synthesis. So if you recall, DNA polymerase can add nucleotides onto a 3' end. The problem that we have is at the 3' end of our chromosomes, and remember there'll be a 3' end on each side, there is no sequence to lay down an RNA primer to then attach the nucleotides to. So at minimum, we would start with a little primer here and then start adding DNA in over here. The problem there is that when we come in and then degrade the RNA primer, DNA polymerase can't come in and fill that gap. So regardless, every single round of replication, the ends are going to get shorter and shorter and shorter because there is no template to put a PCR primer on. So this is a problem because over time our chromosomes would become progressively shorter. And in fact, this still does happen. The cells try and solve this problem through a specialized mechanism catalyzed by the enzyme telomerase. Now, telomerase is a protein RNA complex, and essentially what happens is this protein has its own RNA template that is complementary to some of the DNA sequences found in the telomere repeats. So essentially, telomerase solves this problem by having its own template. So this is better understood in a diagram here. So here we have a 3' end, and what we need to do is start filling in a sequence over here, but we need some piece of RNA to add a 3' end to. So what telomerase does is it has a sequence internal that is complementary to this telomere sequence in part and contains the complementary sequence for the rest of it. So it will bind to the 3' end. It will add on nucleotides to the end, and this can happen over and over and over again. So here we've added on here. We just shift it over, add on more, shift it over, add on more. Now when primase comes in and sits down, it's sitting down over here, creating this RNA primer, and we still have the problem that we can't actually replicate to the very end of this 3' end. But what we've done is put on repetitive sequences at the end over and over and over again, so that when we do lose DNA, it's only the repetitive sequence, not an actual gene. So as we mentioned in class, there is a relationship with telomerase activity in both aging and cancer. So as we get older, we have when we're born, we have very highly active telomerase. As we get older, our telomerase activity decreases over time. And for a lot of cells, that is not an issue. A particular muscle cell doesn't really have much need to divide. And when we do need cell division, what we can do is maintain telomerase activity in stem cells, and the stem cells can stay active and replace any cells that are needed. But our fully functional cells don't need to divide anymore. So consequently, they lose it, or they lose telomerase activity. This will lead to a shortening of telomeres. So at birth, our telomeres are about 8,000 base pairs in length. In an elderly person, they can get as 1,500. Now, the problem that occurs is as the telomeres get to a certain shortness, the cell preemptively turns off its ability to divide, or becomes what we call senescent. And this will help protect the cell, because since it has too short of telomeres, if it doesn't divide, nothing bad can happen. Now, if you were to take a cell that was getting close to senescence and somehow reactivate telomerase, this would block this senescence process. So telomerase, if we could reactivate it, would prevent cells from becoming senescent. And while this might have some benefit in aging, it also carries a risk, because one of the issues that cancer has to overcome is its ability to reactivate telomerase. So in order for cancer to occur, and again, cancer is just simply a cell in your body that starts replicating outside of controls. So it's essentially a runaway cell. And it loses the ability to both take directions from other cells in the body, as well as recognizing what kind of cell it should be and where it should be living. But one of those things that it needs to overcome is its activity of telomerase. Since cancer is rapidly dividing, it needs telomerase to be fully active. Consequently, telomerase is a target for anti-cancer drug treatments, right? So if we want to stop cancer, if we turn off telomerase, eventually that cancer will stop dividing. Now, this type of treatment would not come without a toll on your body. So essentially, when we are targeting cancerous cells, we are targeting specifically the fact that they are rapidly dividing. When we induce treatments that will help stop that, be it against telomerase or some other target, We also tend to target any other cell in our body that is rapidly dividing. And we do have some of those, right? So our hair is constantly growing, our skin is constantly replenishing, and our immune system is constantly generating new cells. So, what happens when you go on anti cancer treatment? Well, you start losing your hair, you develop skin problems, and you have a very weak immune system. So, you can see that while this is a good target, it doesn't come without consequences. And on the flip side, if we wanted to fight aging, keeping telomerase active longer would help, but it increases the risk of cancer. So it's kind of a double-edged sword.